this video, we're going to be extending our discussion of genetic inheritance and starting with looking at genetic inheritance in humans and the depiction of those inheritance patterns using human pedigrees or family trees. And then what we're going to do is we're going to talk about other non-Mendelian inheritance patterns that get into some of the more complex patterns of inheritance that have been discovered since Mendel's time. But let's start with our discussion of analysis of human genetic traits. So to begin, I just want to remind you of a basic scientific principle that we've talked about in the course before, but is particularly applicable to pedigree analysis, and that's the idea of parsimony, looking for the simplest possible explanation for the phenomenon under observation. In this case, the phenomenon that's under observation is the pattern of inheritance depicted in a family tree or observed in a family tree. And there's a great quote that comes from Dr. Theodore Woodward, who's a professor at the University of Maryland School of Medicine, that he gives his medical school interns in The Art of Diagnosis, that says, when you hear hoofbeats behind you, think of horses, not zebras, right? Occam's razor tells us that we need to be looking for the simplest, most likely explanation for the pattern that we're observing. So in pedigree inheritance questions in genetics, in this class or in your genetics class, when you're asked to determine the pattern of inheritance, what we really want to do is answer two questions. We want to answer the question of, is the trait inherited in a dominant or recessive fashion, and is it autosomal or sex-linked? Now remember, what we mean by these terms autosomal or sex-linked is that our autosomes are chromosomes number 1 through 22, and sex-linked refers to our sex chromosomes, which are the X or the Y. Right? And so as we're going to see, inheritance of traits on the autosomes is exactly the same for males and females. Both males and females have 2 of each of these types of chromosomes. However, where it gets different is in the sex chromosomes, because we know that females have 2 X chromosomes, whereas males have an X and a Y, and so therefore they have only one X and one Y. So what we'll see is a difference in inheritance patterns that are associated with genes that are located on the X or the Y chromosomes, whereas those differences between males and females don't exist for the autosomes. So these are the two questions that you need to answer when you're asked to determine a pattern of inheritance in a pedigree. So let's take a look at some pedigrees and see how we can determine patterns of inheritance by looking at pedigree genetic trait inheritance. So first of all, we need to know what we're looking at in a family tree like this. So when you're looking at a tree like this, any two individuals that are connected by a cross line are individuals who've had a child together, or more than one child together. Females are depicted as the circles. Males are depicted as the squares. Anybody who's shaded in the pedigree is showing the trait that you're investigating. Generations start at the top with the oldest generation, and then the younger generations are listed top to bottom. And we identify the individuals in the pedigree by number. And so first we number the generations from top to bottom, oldest to youngest generation. So the top generation here would be generation 1, second generation down would be generation 2, and then generation 3 here at the bottom. And then in each generation we number every individual who was either born into the family or married into the family and has offspring, left to right as so. And so if we wanted to specifically refer to this individual in the pedigree, this individual would be individual 3-4. All right? Okay, so shaded individuals show the trait, not always a disease. Maybe we're looking at something like freckles or some other innocuous trait, but a lot of times when pedigrees are being analyzed, it is in the context of trying to understand how a disease that affects humans is passed on across the generations. So when we're looking at a pedigree, we need to be thinking about the inheritance patterns that we saw previously in our discussions of monohybrid crosses initially. Remember that we said that when we have two alleles combined in an individual and we see the expression of one of those alleles over the other, then that is a dominant allele. So for example, with Mendel's peas, we looked at flower color where we had a dominant purple allele combined with a recessive white allele, right? That was a heterozygous individual, and in that individual plant we would see what phenotype? We saw purple, which tells us that the purple allele is dominant over the white. And so for any dominant trait, and this applies no matter what organism you're looking at, having even one of that dominant allele overrides the expression of the other. It doesn't matter if that other allele is the dominant allele or the recessive allele, you get the same phenotype associated with that, right? So either homozygous dominant or heterozygous, both equal purple. Same is true for genetic characteristics in other organisms like human. So what this means is that if you show the dominant trait, then you have this genotype. You have the dominant allele and anything else, right? And if you have even one of that dominant allele, the corollary is you're going to show the trait. So when we're evaluating pedigrees, we need to think in those terms, right? For a dominant trait, having even one of the allele means that you're going to have the trait. But what about for a recessive trait? For a recessive trait, you actually have to have two recessive alleles. So for Mendel's flower color example, we had the recessive trait of white flower color, and the only way that you were white flower colored was if you had two recessive alleles, if you were homozygous recessive. Well, same exact thing applies for human genetic traits that are inherited in a recessive fashion as well. All right, so how does this apply to a pedigree? Well, we need to think about parents, all right? So I always recommend that students start with an affected individual and then look at that affected individual's parents and work their way backwards in the pedigree. If we're talking about a dominant trait, and you are affected by that trait, let's say it's a disease, you're showing this dominant disease. If we look at your parents, is there any possible way that both of your parents could be unaffected? In other words, that they might have the mutation but not be showing the mutation? And the answer is no. Because if you inherited that dominant allele, where did you get it from? You got it from one of your parents, right? This individual had to have at least one of their parents with that dominant allele. Otherwise, you know, we're talking about maybe the milkman is the parent or something, because this individual had to get that allele from somewhere, so one of his parents had to have it, or her parents. And if that parent had the allele, then they must also show the trait. So if a trait is dominant, any affected individual is going to have an affected parent as well, right? Every affected individual has an affected parent. The same is not true for a recessive trait. Think back to Mendel's experiment. How did we get in that F2 generation individuals showing up with those white flowers? Well, it was because two heterozygous individuals had been crossed to one another, and if we look at that pedigree, we know that we expect 1/4 of the time that we're going to end up with the homozygous recessive phenotype and genotype, right? We're going to end up with the homozygous recessive genotype, and that's going to be associated with the recessive phenotype. Now, if we look at the characteristics of these two parent plants, though, neither one of them was white. They both carried a silent copy of the white allele, but neither one of them was showing that trait. So we might refer to those as silent carriers of the white allele trait. For recessive traits, you can have silent carriers. In other words, you can have an affected individual with unaffected parents. In a dominant trait, you can't have that. No silent carriers are possible with a dominant trait. So how does this play out in a pedigree? If you see a human pedigree that has a characteristic pattern like this, and we see that every individual who's shaded in, if we look at that individual's parents, one of their parents also shows the trait, that's a highlight of or a hallmark of a dominant trait, dominant acting trait. So what would a recessive trait look like then? Well, here we could see affected individuals showing completely unaffected parents. All right. So these two individuals, this male and this female, in the fourth generation here, both show a trait that neither of their parents exhibited. It's not possible for this to be a dominant trait, because if it were dominant, they would have had to have inherited that dominant allele from one of their parents, and that parent would have to show the trait. Right? So this is the sort of analysis we're going to be doing. Now in this pedigree we can actually see a new notation showing up, which is a double crossbar connecting two individuals. And what this represents is what's called a consanguineous union. And consanguineous means with blood. So this is a union between two individuals that are to a greater or lesser degree related by blood. So in this case we can see that they're first cousins, right? Now in the Western world this is not as common as it was, say, maybe 100 years ago. It's very much frowned upon. But in other parts of the world it's still really, really common to have matings between particularly second cousins and beyond. And this consanguineous mating can be associated with an increased risk for recessively inherited disorders. And the reason for that is this: every single one of us in the population is carrying, somewhere between maybe 5 to up to a dozen alleles for recessive diseases. If you got your genome sequenced, you would see that you were a silent carrier for somewhere between 5 and a dozen recessive disorders. Now, if you're going out and you're mating with other individuals in the population at random, it's very unlikely that you're going to be running into an individual that just so happens to have your particular set out of 20,000 genes of recessive alleles, right, for any one of those. But if you're mating within a smaller population of individuals that are more closely related to one another, say a small village is geographically isolated or something like that, then the likelihood that you're going to be mating with someone who shares any genetic liabilities that you have is going to increase. And so a lot of times when pedigrees for recessive disorders are analyzed, if we're looking at other parts of the world, you will see these kinds of consanguineous matings that have resulted in an increased risk of having homozygous recessive offsprings that show the trait, whereas the parents were just carriers of the trait. Okay, so recessive traits, silent carriers may be present, right? Dominant acting traits, no silent carriers are possible, because if it's a dominant trait, if you have even one of that allele, you're going to show the trait. Okay, so that's how we distinguish between the first question here, dominant versus recessive. We're looking for the presence or absence of silent carriers, essentially. What about this business of autosome versus sex chromosome? Okay, so first we need to understand something about sex-linked inheritance and sex determination. So the X chromosome has about 2,000 genes on it, most of which have nothing to do with sex or secondary sex characteristics. Some of them do. Estrogen receptor is an example of a sex characteristic associated gene that's present on the X chromosome, but most of the genes on the X have nothing to do with sex determination and really are just involved in normal physiology. So this is a giant chromosome, tons of genes that are critical in their function for both males and females. The Y chromosome by comparison is tiny, and it only has about 70 functional genes, right? So less important in terms of overall physiology. One of those genes is critically important for sex determination, and that's the sex determining region on the Y gene, or what's called SRY. This SRY gene is responsible for kicking off the developmental events that lead to the differentiation of a developing embryo from the default female pathway to the male specific pathway, right? So early in human development, the default embryonic state is more or less female. And if the SRY gene is present, i.e. if the Y chromosome is present, then the developmental pathway takes a sharp right turn, and all of the structures that would have undergone the female-specific developmental pathway degenerate, and instead we see the promotion of the development of male-specific reproductive structures and male-specific gene expression starting to be initiated, right? So if SRY is present, you get the characteristic of being male. If it's absent, then you get the characteristic of being female, right? So as we said before, females are XX and females are XX and males are XY, all right? So what is the consequence of this? Well, where we see this really playing out is with recessive alleles. So remember, under ordinary circumstances, if we're talking about an autosomal gene, the only way that you're going to show a trait for a recessive autosomal gene is if you inherit two bad copies, we might refer to them as, of that recessive allele. Why? Because if you inherit even one of the dominant allele, that's going to be associated with the dominant phenotype, right? So for autosomes, the only way you show a recessive trait is if you are homozygous recessive. But what about recessive traits that are on the X chromosome, that are encoded by genes on the X chromosome? Well, now we have a difference, because females can have a backup copy of the X chromosome that has a good version of whatever that gene is. So this sort of scenario, right, where they could be silent carriers for a trait on the X, males don't have that option because they only have one X chromosome. And so if males inherit a recessive allele on the X chromosome, they have no other X chromosome that could potentially mask the expression of that trait. So what this leads to is that males will express recessive X-linked genes very easily, whereas females will express them no more easily than they would a regular autosomal trait. Okay? Now, how does this play out in a pedigree? Well, this is what that looks like. If we're talking about a recessive allele, then affected individuals are almost always going to be male. And who are they going to be inheriting this recessive allele from? Well, not dad, because dad is passing on a Y chromosome to his son, right? Dads pass on Y chromosomes to their son. Mom is responsible for passing on the X. And so if these sons are inheriting an X chromosome that has a defect, they're inheriting it from their mom. Now, is mom going to show the trait? Probably not if it's a recessive trait, because mom will probably have a heterozygous genotype where she's only going to have one bad X and one good X, and so she will be a silent carrier for whatever this X-linked trait is. But in passing this bad X on to her son, he now only has one X chromosome and he's going to show this trait, whereas mom didn't. All right? So hallmarks of recessive X-linked inheritance, and when we say sex-linked, we almost always are talking about X-linked because there's so few chromosomes on, so few genes on the Y chromosome. So number one hallmark, mostly males are affected and they're inheriting through their moms, right? You can see both of these males are inheriting from mom. Now, the other thing is that if we do see a female, so if we had another pedigree, let's say where we saw a man and a woman, they had offspring, and we saw one of their offspring was female. If this was an X-linked trait, and she would have to be homozygous, recessive for that trait, right? She'd have to have two bad X's. Well, where did she get her X chromosomes? One would have had to come from mom. The other would have had to come from dad. And if dad has a bad X because he doesn't have a backup Y, he is also going to have to be affected by whatever this trait is. So in X-linked inheritance, if there are affected females, they have to be homozygous for that bad X, which means that their fathers must have also been affected. If that's really her dad, she had to get a bad X from him. All right? Now, here's a question for you to consider. This is with reference to recessive traits, which can be masked by a dominant allele on the other chromosome. Would dominant traits differ in their pattern from autosomal dominant traits? So would X-linked dominant traits be inherited in a different fashion from autosomal dominant? And if so, how? If not, why not? So think about that, and we'll talk about that more when we have our genetics problem-solving activities in class. Okay, so that's the basics of inheritance patterns as depicted in pedigrees, and how we distinguish between autosomal versus sex-linked, and how we distinguish between dominant versus recessive. Let's move on and talk about some other non-Mendelian patterns of inheritance that Mendel didn't observe, but which we now know exist out there in the natural world. The first one that we're going to be looking at is what's called polygenic or quantitative inheritance. And what this refers to is the interaction of multiple genes all contributing to one phenotype. And as you can imagine, there are a lot of phenotypes that are determined by the interaction of many different genes that all have to contribute. So if we think about something like height in humans, you could imagine that that's going to be dependent on a whole host of factors that are genetically determined. So you're going to have growth hormone that influences the height at which an individual is going to finally be determined to be. You're going to have issues with regard to bone growth and development. You're going to have issues with regard to nutritional status, ability to absorb nutrients, and how efficiently you put those nutrients to use, mineralization of bone, things like that. So lots and lots of different genes, as you can imagine. So there are a lot of traits that are like that. Probably, in fact, more traits are polygenic than not with regard to human physiology, and that's true across the natural world as well. So how does this work? How can we see this playing out in terms of inheritance patterns? Well, let's take an example from plant. So this is wheat seed color, which is determined by 3 separate genes A, B, and C. So these are different genes, different loci in the genome, each of which is responsible for conferring a little bit of redness to wheat seeds. All right? So everywhere we see a dominant allele at the A, B, or C gene, that confers a little bit of redness. So let's imagine that we have 2 true breeding strains of wheat, one of which is very dark red, and if we look at the A, B, and C genes, we see that that's because they are homozygous dominant at those 3 genes. The other parent has white wheat seeds, right? And if we look at their A, B, and C genes, we find that they are homozygous recessive at all 3 genes. In other words, they have no alleles conferring redness. And so their seeds are white, right? All 6 alleles that are possible for this other parent are conferring redness, and so their seeds are going to be dark red. If we cross these 2 wheat parents together, then what are we going to get in the offspring? Well, if we think about the gametes that are going to be produced, this parent can only produce one type of gamete that has a dominant allele at each. This parent can only produce one type of gamete that has a recessive allele at each. And what we would expect to see in our F1s are individuals that are heterozygous at all 3 genes. So an intermediate phenotype, halfway between the 2 parents, pink, let's say, right between them. Well, so far this kind of looks like incomplete dominance, right? And the way that we would be able to distinguish this from incomplete dominance is in the next generation. So if we allow these F1 individuals to self-cross or to intercross with one another, now we get some complexity and some genetic variation being introduced into the population. Because think about this. These seeds that are going to be produced by these F1 plants could have ranging from 0 red conferring alleles all the way up to 3 red conferring alleles, right? Because what kinds of gametes are going to be formed by these F1s? Well, again, we could use a branch diagram to depict all of the gametes. So we know we're either going to have a dominant or recessive A allele, dominant or recessive B allele associated with each of those, and then dominant or recessive C allele associated with each of those. be a capital. And if we follow the branches down, we can see that we've got these possibilities for gametes. So capital A, capital B, capital C, capital A, capital B, lowercase c, capital A, lowercase b, capital C, capital A, lowercase b, lowercase c, and so on. You can do the rest of the branches. But we end up with these 8 types of gametes that are possible. Because both the female and the male plants are genetically identical, we've got 8 possibilities down both sides, and so we end up with this 8 by 8 Punnett square where we combine by fertilization gametes from the male, gametes from the female, and every possible combination. And what we're seeing here is counting up in the offspring that are produced by those fertilizations how many red conferring alleles that offspring is going to inherit. So you can see that this offspring up at the top of our diamond is getting 3 red alleles from this parent, none from this parent, and so they have 3. And they end up with an intermediate pink phenotype that looks just like our F1s. These individuals that only inherit 2 red alleles are going to be slightly lighter in color. These individuals that only inherit 1 allele from this parent, this one's giving nothing, would be lighter still, and these individuals that inherit 0 red alleles from the 2 parents would have no redness. They would be white, just like this original parent we started with. On the other end of our box, we have the possibility of getting 3 red alleles from this parent and also 3 red alleles from this parent, which would give us 6 red alleles, and they would have the darkest seed color of all, which would match that dark red of the original parent, right? So we get a whole spectrum of colors that range between the 2 original parental colors of white and red and every shade in between. Right? So we get a variation, a spectrum in shading between our 2 parental strains, not just this intermediate color, but every shade in between. That intermediate color is going to be the most common because there are more ways for us to produce plants that have 3 alleles than there are ways to produce plants that have only 1 or 2 or 0, right? And so the same thing going on in this direction. 4, 5, and 6, those are rarer combinations in our offspring that we expect to see. So our intermediate phenotype is going to be the most common in our F2 generation, but we will see all the other shades present. And so we get this histogram that shows this bell-shaped curve with most individuals lying in the middle and then fewer individuals tailing off on either end. And if we look at many traits associated with humans, so everything from height to weight, and look at eye color, as we'll see in just a minute, disease traits like diabetes, susceptibility to diabetes, high blood pressure, cancer, Alzheimer's, severe anxiety, depression, we would see the same sort of influence of multiple genes, and included with that would be some environmental effects as well, obviously. But most of these complex diseases that we see in humans are influenced by multiple genes, not just one gene, one trait. So that's polygenic inheritance. Here's a nice depiction of the vast range of human eye colors. It's very often sort of simplified as, you know, brown eyes versus blue eyes versus green eyes, and brown is dominant over blue, and blah, blah, blah. But that's actually a vast oversimplification of the genetic determination of eye color, which relies on many different genes that determine not just what types of pigments are going to be made, so do you make brown pigment or not, but the amount of pigment that gets made, the pattern in which it gets distributed in the iris of the eye, other structures that are present that may scatter the light being reflected from the eye in different ways. So there really is a huge spectrum of colors that are possible because there are so many genes that are involved with determining the aspects of physiology that lead to that overall phenotype of eye color. All right, so let's move on. So let's talk about a second non-Mendelian pattern of inheritance or influence on inheritance that we see, and that's environmental effects. So Mendel didn't describe this, but it should be pretty obvious that it's been apparent for a very long time that many traits, even those that are genetically determined, in other words, there's a gene in the genome that determines the phenotypic expression, are going to be influenced by environmental effects as well. Think about buying a packet of tomato seeds that are genetically identical to one another and planting them in your garden, and planting some of those seeds in very fertile soil, planting some of those seeds in very nutrient depleted soil, varying things like watering patterns. You could see that you're going to get very different phenotypic expressions from those genetically identical seeds that you've planted, right? That makes sense. Same thing is true for other genetic traits as well. So a good example of that is seen in humans, where we have this trait of what's called male pattern baldness. So this is to differentiate this pattern of baldness from the normal age-related baldness that happens to absolutely everyone, which is baldness that happens over the head as a whole, so thinning of the hair overall. That happens to everyone. But in male pattern baldness, we see a very different pattern of baldness that affects the top of the head specifically. So the sides are left relatively intact for a longer period of time, and primarily what's affected first is the hair up on the crown of the head. And so we see this very characteristic thinning that occurs from both directions, front and back over the top of the head, leaving this monk-like distribution of hair. So the cool thing about this is that it's influenced by environment in such a way that testosterone levels in the body can influence the expression of this trait. Individuals who have high testosterone have earlier onset of male pattern baldness than individuals who have lower testosterone levels, which is a really weird thing if you think about it, because what that's saying is that the hair follicles that are specifically located here on the top of the head are influenced by testosterone levels in a way that the hair follicles on other parts of the head and the rest of the body are not. And we know very little about how that's controlled, but we know that it's absolutely true. And so the phenotypic expression of this trait is actually a combination of both genetics and environment that these cells are experiencing with regard to their testosterone exposure. All right. Now, the other cool thing about this, and this is something that wasn't known until fairly recently, is that this is not a single gene trait. There are actually multiple genes that have been identified that influence the onset of male pattern baldness, and one of those genes is on the X chromosome, so it's sex-linked as well. So this is one of the most complex traits that we could imagine. It is polygenic, multiple genes. One of them is X-linked, so we've got X-linked expression as well, and it's environmentally influenced. All right, what else can we see? So let's talk about two additional non-Mendelian inheritance patterns or phenomenon that we see, epistasis and pleiotropy. And in order to talk about these, we're going to look at coat color in cats, which is a really interesting genetically determined trait. Actually, coat color, genetic determination of coat color in other animals is well described as well, and it's a really interesting phenomenon that sees expression of multiple genes in different patterns to determine the overall phenotypes. We can look at the same thing in dogs, we can look at the same thing in horses or mice, and we'd see similar phenomena at work. So first thing we need to know, as you guys are all aware, cats come in many colors, and the coat color is possible to be observed in many different patterns as well as shape of the coat and the texture of the fur, and curly versus non-curly, and long fur versus short fur. So there are lots of different genes that determine coat color, but a small subset of those, just determining the color pattern specifically, we have genes that determine whether black fur versus orange or red fur is going to be seen. We have a different gene, so this is a completely separate gene, that determines whether that color is going to be diluted or not. So in other words, a cat that has a gene for black fur might have an allele at the separate gene that causes that color to be diluted in such a way that we see a gray cat instead of a black cat. There's yet a third gene that determines the pattern of color distribution, so striped versus not striped, and a fourth gene that determines whether the pattern observed is going to be spotted, so whether we'll see spotted areas lacking pigmentation versus not spotted or solid color. So the overall coat color that we see associated with a cat is determined by multiple genes and the alleles that we would see, the genotypes that we would see at each of those genes, right? So this cat obviously has an allele at this first gene that encodes black color. At this second gene that color is not diluted, so it has a not dilute allele. At the stripy gene, it has a version that says no striping, and at the spotted gene, it has a version that says no spotting. And so the phenotype that we see overall is a solid colored black cat, right? That makes sense. So we could look at all these different cats and their coats and figure out what alleles they would have to have at these different genes that's resulting in that overall phenotype. There is a different gene, the W gene, where a specific allele, the white allele, is what's called epistatic to all of the other coat color genes, which means that it overrides or blocks the phenotypic expression caused by those other genes. So in other words, that previous cat that we just saw, this black cat that had a solid coat, if this cat had inherited this dominant acting W allele at the white gene, it would not be black. Instead, it would be white, right? Same for all these other cats. It wouldn't matter whether they had the black versus orange allele, whether they had the dilute or not dilute, striped versus not striped, spotted versus not spotted. If they inherit this specific dominant allele at the white gene, then all of those other coat color genes' expressions is blocked. Now, how can that occur? How can we get epistasis or overriding of one gene's phenotype by another gene? Well, typically what we're looking at is a gene that functions upstream in a developmental or metabolic pathway. And in this case, the gene that we're seeing that's overriding the expression of those other coat color genes is one that is critically important for the development of a cell population called melanocytes, which we've talked about before in our class when we talked about PPO initially. Melanocytes are the pigment producing cells, right? So they make pigment. So that's what determines what color skin, what color hair you're going to have. Well, it turns out that in order to have these melanocytes developed at all, we have to have no expression of this dominant W allele. In other words, having this dominant W allele knocks out the melanocytes in the body. So they never get produced at all. So if you don't have the cells that produce the pigment, obviously you're not going to have any pigment present. No melanocytes, no coat color, regardless of whether those cells would have been producing the black or orange, dilute, not dilute pigment colors, right? So this is epistasis. An allele of one gene overrides the phenotype caused by a different gene. And that's very important to recognize. We're talking about two different genes, right? The white gene overriding expression of the black versus orange gene, for example. This is not simple dominance and recessive interaction. This is one gene blocking the expression of a completely different gene because it's operating upstream in the pathway. So that's epistasis. This coat color inheritance also demonstrates another interesting pattern, and that's what's called pleiotropy. Pleiotropy is where one gene has more than one phenotypic effect, right? More than one phenotypic effect. And the way that we see this playing out with the W allele of the white gene is that cats who inherit that W allele are not only white, lacking pigmentation because they lack melanocytes, but they're also usually deaf. And the reason that they're deaf is that within the inner ear, there's also a population of cells that are related to melanocytes that regulate the balance of electrolytes within the ear that controls action potentials and the transmission of electrical signals to the brain. So if we don't have melanocytes in the inner ear, then we get no electrical transmission. We end up with deaf cats. And there are other phenotypic outcomes that are associated with that W allele, but these are the two that are most common. The white pigmentation, loss of pigmentation because we have no melanocytes present in the skin or in the hair follicles, and then also inappropriate development and behavior of the inner ear and loss of the ability to transmit electrical signals to the brain. So pleiotropy, one gene has more than one phenotypic effect. Now, very often students will confuse the phenomenon of pleiotropy with polygenic, right? These are two sides of the same coin. Pleiotropy refers to one gene having multiple phenotypic effects, right? So one gene causing phenotype 1, phenotype 2, phenotype 3. That's pleiotropy. With polygenic inheritance, what we're talking about instead is multiple genes all converging on one phenotype, right? So they're sort of dealing with multiplicity in opposite directions here, all right? But that's pleiotropy. Okay, it turns out we have the same gene, or very closely related gene in us. It's called the KIT gene in us, not the W gene, but it's the same gene, and it's doing the same thing. And mutations in that gene have been identified in humans and in experimental models, mouse, and those recessive mutant alleles act the same way that we see the W allele behaving in cat. So if you're heterozygous for this KIT gene and you have one recessive allele and one normal dominant allele, then we see defects in pigmentation that are referred to as piebaldism. So we have this characteristic patchy pigmentation where you're missing pigmentation on the belly, missing pigmentation on the forehead, in this very characteristic pattern. And the reason that we get this pattern is because we have a defect in the migration of the cells that are going to become the melanocytes, which normally come from the back of the body where the neural tube is early in development and migrate around and then come together at the midline, right? So normally we have migration that looks like this around the embryo and meeting up at the midline. And for whatever reason, melanocytes never make it to these two patches of epithelial tissue, and so we have a lack of pigmentation there. Interestingly, you see exactly the same phenotype associated with that heterozygous genotype in mouse, and it really does demonstrate that humans are basically just giant hairless mice when we see these kinds of similarity in phenotypic expression. If you're homozygous for that recessive mutant allele, you actually have much more severe pigmentation problems, so you do show the albinism that would be more similar to the dominant allele that we see in cats, deafness as well as sterility, and defects in the enteric nervous system, which is the nervous system that is regulating the gastrointestinal tract and its function. So those individuals have various defects with the ability to digest and absorb nutrients across that GI tract. That same gene has other alleles that have arisen by random mutation that are dominant acting, right? And those dominant acting alleles are associated with various forms of cancer and increased susceptibilities to those cancers. Now, why would that be? Why would we have a dominant acting allele associated with cancers? Well, it turns out that what this gene encodes is a receptor tyrosine kinase. And this specific receptor tyrosine kinase has as its ligand a growth factor that stimulates cell division of stem cells that are going to form those melanocyte populations as well as many other populations in the body. And so what the dominant mutations do is hyperactivate these receptors so that they are less dependent on those ligands binding, and they can actually signal inappropriately and in excessive fashion to the cell, triggering excessive cell division. And so we see these types of mutations in various types of gastrointestinal cancers, testicular cancers, melanomas, leukemias, as well as many others. But this is an excellent illustration of our ability to use mouse models to study genetic disorders that are very, very relevant to humans. Okay, so we already addressed polygenic and pleiotropic. Okay, so moving on, let's talk about another non-Mendelian inheritance pattern that wasn't described by Mendel, but which we now know exists, and that's X inactivation. So going back to our discussion of sex determination, we already talked about that having two X chromosomes leads to the determination of sex as female, right? And having one X chromosome leads to determination of sex as male by virtue of the fact that that Y chromosome has an SRY gene that causes that developmental pathway to occur. But if we think about this, this actually presents a problem. We've discussed before that for most of our genes, we have to maintain appropriate gene dosage, and we don't tolerate deletions or extra copies of chromosomes very well at all, because we end up with gene dosage effects. So here we have an example of females having double the genes for all of those, some 2,000 genes on the X chromosome compared to males. So how is that possible without having defects? Well, it turns out that in females, what we do is randomly in every cell of the body early in development take one of those X chromosomes and coil it, condense it down, compact it so that it's not expressed, or almost entirely not expressed, with the exception of a few genes that escape X inactivation. So if you look at cells from a female individual, you can actually see within the nucleus of the cell these little highly compacted and condensed structures that are actually one of the X chromosomes that's been coiled up and condensed even more tightly than it would be during mitosis, and that gets segregated to the side of the nucleus and sort of pushed up against the membrane. So this is X inactivation. So in doing so, what we do is compensate dosage between females and males, reducing the dosage of X-linked chromosomes in females to match that of males, right? So this is dosage compensation. We take one X chromosome, compact it, silence it, and reduce females' gene dosage so that it matches that of males. Now, other organisms do this dosage compensation differently. If we looked at flies, flies are also characterized by XX being female and XY being male. But rather than having X inactivation, what flies do is they take that single X chromosome and they double the expression of every single gene on their single X, bringing it up to a level that matches that of female, right? So there are different ways to accomplish dosage compensation. X inactivation is just the way that it's accomplished in mammals like us. We can see interesting phenotypic effects that arise from X inactivation when we have heterozygous genotypes for genes on the X. So a nice example of this is calico cats. It turns out that that black versus orange pigmentation pattern is determined by a gene on the X chromosome. And so we have two alleles. One allele confers the ability to make black fur. The other allele confers the ability to make orange fur. Early in development in every cell of the cat's body, if this is a female cat, one of those X's or the other is going to be compacted, condensed, inactivated, and you're only going to see expression of the other allele. So in all of the daughter cells that are produced by this cell's lineage, we're going to end up with a cat with areas of the coat that have orange fur, and in all of the descendants of this cell, we're going to see that black fur being formed. And so you get this overall calico pattern that's associated with the coat, and oftentimes this is combined with the spotting allele at the spotted gene, and so we'll actually end up with some white patches occurring as well. So calico cats are almost always female. The only way you get a male is if you actually have an XY male, and we see that kind of inactivation pattern producing that calico coat distribution pattern as well. Does this have any implications for humans in terms of clinical relevance? Well, not too many examples have been identified in humans, but there are a couple of genetic disorders that are associated with this kind of chimerism in terms of which X chromosome is being expressed over the other. And one of those has to do with the development of sweat glands, which is controlled by a gene on the X chromosome. And so if you have a mutation in that gene and you're they're heterozygous, depending on the distribution of X inactivation, you could actually end up with patches of skin in your body that don't have functioning sweat glands. And if the areas of the body that don't have those functioning sweat glands happen to be in parts of the body that are particularly important with that evaporative cooling function that's associated with the sweat glands and production of sweat, then you can actually end up with a defect in regulating temperature through sweating. And so that's one of the more common ones. But there are probably others that haven't been identified yet, or that are less apparent. All right, so that's X inactivation. Moving on, let's talk about something else that's related to females, and specifically female gametes and the way that they contribute to the next generation of progeny. And this is mitochondrial inheritance. So by a quirk of the way that the gametes come together and what they contribute to offspring, it turns out that that mitochondrial genome that we talked about in lab, every single mitochondrion having its own genome and its own, small set of genes that are critically important for electron transport chain function and other functions in the mitochondrion. Turns out we only inherit that mitochondrial genome and the mitochondria as a whole from mom. And the reason for that is because of a difference in the way that the gametes develop in males and females. The female gamete, the egg, is a large cell, contains lots of cytoplasm, lots of mitochondria. The male gamete, or sperm, essentially during the process of development extrudes almost all of the cytoplasm and organelles, and really just becomes a genetic payload in the head of the sperm. And when we see fertilization or fusion of sperm and egg, what's delivered is this genetic payload into the interior of the oocyte. So we see the DNA being transferred in, and there are very, very few paternal mitochondria within the head of the sperm at all. And if we just so happen to see some of those mitochondria make their way into the egg during the process of fertilization, the egg would actually enzymatically destroy those mitochondria. So what that means is that every fertilized embryo has acquired its entire complement of mitochondria from mom, not dad. And so if we're thinking about the inheritance of defects in mitochondrial genes, if you have a mutation, let's say, in one of those electron transport chain genes, you can't inherit that mutation from your dad. You can only inherit from mom. And this is referred to as a parent of origin effect. Because you don't inherit any mitochondria from dad, you can't inherit any mitochondrial mutations from dad. So that's one thing that we see with mitochondrial inheritance is inheritance from the maternal germline, but not the paternal germline. Now, the other thing that we see, because of the fact that the distribution of mitochondria during cell division is essentially random, whatever mitochondria end up on this half of the cell are going to go that way during cell division, and whatever mitochondria end up in this half of the cell are going to go that way during cell division. We can have variable levels of inheritance of the mutation in the mitochondria that carry those mutations themselves, right? So these little red spheres are meant to depict mitochondria that have the mutation, and as we can see in this depiction, just by random chance, this daughter cell that's being formed during cell division is inheriting more mutant mitochondria than this daughter cell. And so we might expect that if that's a deleterious mutation in the mitochondria that's causing a negative impact, then all of the daughter cells that are produced from this cell are going to be negatively impacted, and the daughter cells from this cell are going to be less negatively impacted. So we can have a varying level of inheritance of the mutation itself, and therefore a varying level of phenotypic expression depending on which cells in the body actually inherit the mutant mitochondria. And this is referred to as variable penetrance of the phenotype. And so depending on how mitochondria segregate during cell division, you could end up with most of the cells in the body showing mutant mitochondria and being affected by that, or the converse. You could have most of the cells in the body not showing mutant mitochondria and not being affected by it. It's essentially random as to how those mitochondria segregate during early development. So how would this look on a pedigree? Well, we would see both of those phenomenon showing up in a pedigree, both the parent of origin effect, where you can't inherit from dad, but you can inherit from mom, and the variable penetrance phenomenon as well. So here we're seeing mitochondrial inheritance, and what the half shading is meant to depict is someone that you can, using genetic technologies like PCR, detect the mutation as being present in their body in some of their cells, but they're not showing a phenotypic expression of the mutation. So they're carriers, they have the mutation in a limited population, but they're not showing the trait. And so that's variable expression of the phenotype. Just by bad luck, the way that mitochondria segregated during development, these other individuals ended up having more of their mitochondria impacted to a greater extent, and so they show the phenotype to a greater degree. They are affected by that trait. And you can see all kinds of spectrums of levels of penetrance in between those two extremes. And here's the parent of origin effect that we talked about. So clearly this mom had the mutation. She passed it on to her offspring. Both her son and her daughter inherited mutant mitochondria from her, and we see that this daughter subsequently then passed that mutation on to her offspring via the mitochondria that were present in her gametes. This son, on the other hand, passed it on to none of his offspring because they didn't inherit any of their mitochondria from this dad. They inherited it from their mom instead, and so since she's normal, they got normal mitochondria. Right, so that's our parent of origin effect showing up in our pedigree. All right, so the final non-Mendelian inheritance pattern that I want to discuss in this video is one that we have briefly referred to before, and that's this idea that some genes are physically located on the same chromosome, on the same bit of DNA. They're actually physically linked to one another structurally on the DNA chromosome. So this is a depiction of human chromosome 1, which is showing just a very, very small subset of the genes that are present. There are over 3,500 genes that have been identified to date on human chromosome 1. And so if we're thinking about the assortment of these genes during meiosis, we would expect that genes that are very close together along the chromosome are going to tend to be inherited with one another, right? Alleles of these genes are going to go together, whereas alleles of genes that are very distant from one another on the same chromosome are very likely to have recombination events at some point along that chromosome that could potentially separate those alleles from one another on the chromosome, right? So when we have linked genes, we need to consider that recombination phenomenon that occurs during prophase 1 of meiosis and the impact that that has on independent assortment of alleles, right? So we take this simplified scenario. We have two homologs here, right? So these are homologs, the same chromosome type containing the same specific sets of genes, one inherited from each parent. So let's say this one was inherited from mom, this one from dad. We know that before cell division, before meiosis, each of these is going to be copied. We're going to have two copies, two sister chromatids of these held together. And we know that during prophase 1, we're going to have recombination between those homologs, with breakpoints or recombination events occurring more or less at random along that chromosome. All right, so we talked about this before, that we're going to have multiple points of crossover, these chiasmata that we observe, where genetic exchange between non-sister chromatids is happening. Well, if we think about this, if we compare genes A and D here, and think about the likelihood that those two genes are going to have a crossover event somewhere between them, we would say that's probably highly likely, right? A crossover event has to occur, or else that meiosis is not going to complete. We talked about that before. It's an obligatory event during meiosis. And so we're very likely to have a crossover event in fact, guaranteed to have a crossover event between two genes that are located at opposite ends of the chromosome. In fact, we'll probably have multiple crossover events between them. On the other hand, if we're looking at two genes that are very closely located next to one another on the same chromosome, it's very unlikely that we would have a crossover specifically right here where we would need a crossover to separate these two alleles from one another, right? So the closer together two gene alleles are on the same chromosome, the less likely it is that we're going to have a recombination event between them, separating those two alleles from one another. The further apart two gene alleles are from one another on the same chromosome, the more likely that we're going to see a recombination event somewhere along the chromosome between those two genes that's going to cause them to be separated from one another. So the consequence of that is that we can use the frequency of recombination to determine the relative genetic distance between two genes and use that to create a genetic map that describes the physical location and the distance between genes along the chromosome. So let's see how that would work. So this is exactly the sort of technique that was used to determine the first genetic maps. And what we're looking at here is the genetic map that was created for Drosophila, or fruit fly. And this was created by Thomas Hunt Morgan's research group. And they used this technique of recombination frequency to map all of those genes to physical locations on the chromosome. This was way back in the early 1900s, so there was no DNA sequencing technology that was available. DNA hadn't even been identified as the heritable material yet. Yet recombination mapping was used to generate genetic maps that, when later on DNA sequencing technology became available, confirmed that this was in fact the order and orientation of these genes along the three major chromosomes and one minor chromosome of Drosophila. So pretty incredible work that was carried out long before technologies were available to fine map genes on chromosomes. So let's look at the way that those experiments were done and how these genetic maps were created by Morgan's lab using Drosophila, or fruit flies. So what we're looking at here are two fruit flies that differ in two genetically determined traits. Body color, so gray or sort of a tan color, being determined by the wild type allele at the B gene, right? And wing shape being determined by a second gene, the VG gene, right? So the B gene is associated with body color, and we have two alleles at that gene. The dominant allele being capital B, which is associated with the normal tan color, and then the lowercase allele being associated with abnormal black body color. And then the second gene that we're looking at here is the VG gene, which is associated with wing shape. And so the dominant allele, capital VG, is normal, oval, wing shape, and the lowercase VG gene, recessive allele, is vestigial, so very small vestigial wings that don't function for flight. So that's the two genes that we're looking at, the B gene versus the VG gene, two alleles at each, one dominant over the other. So tan dominant over black, normal over oval, dominant over vestigial. All right, so these are the two parent flies that we are starting with in our mapping test cross. All right, so just like we can use a test cross to determine the genotype of an individual showing the dominant phenotype, we can do a test cross to determine if genes are linked, and if so, to map them relative to one another and determine a genetic distance between them. So how would we do this? Well, we would start with a fly that's heterozygous for the two genes that we're interested in mapping. So here's a female fly that is heterozygous at the B gene heterozygous at the vestigial gene. So she would have been produced from a mating between two true breeding flies. Let's say that looked like this. So true breeding for the body color and true breeding for vestigial, right? When these two were mated, they would result in exactly the genotype that we see for this fly, heterozygous at both, right? We test cross this heterozygous fly to a male that is homozygous recessive at both the black and vestigial genes. Homozygous recessive, and therefore showing the homozygous recessive phenotype associated with that genotype. Black body, vestigial wings. She, having one dominant allele at each gene, shows the wild type or normal phenotype. Gray body, tan body, and normal oval-shaped wings, right? Okay, so how do we do this test cross? So we cross them against one another, and we know that this individual fly is only going to be able to produce one type of gamete. Every single one of this fly's gametes are going to have the black allele at the black body gene and the vestigial allele at the wing shape gene, right? 100%, because that's all this fly has to pass on. But this fly is going to be capable of producing 4 types of gametes, as we saw before. So we could have the tan allele go with normal wing, or it could go with vestigial wing. The black allele could go with normal wing, or it could go with vestigial wing. And so we end up with 4 types of gametes possible for these 2 genes. And here they are. So if we think about it, 1/4 of the gametes would be expected to have the dominant allele for both. 1/4 would have the dominant allele for the color gene, recessive allele for the wing shape gene. 1/4 would have the recessive allele for the body color gene, dominant allele for the wing shape gene, and 1/4 would be expected to have the recessive allele for both, right? 1/4, 1/4, 1/4, and 1/4. Okay, so if we did this cross, not knowing if these 2 genes, B and VG, were linked or not, what would we expect to get if the genes were on different chromosomes? If they're on different chromosomes, we expect exactly what we just said, right? 4 gamete types produced at random, present at equal proportions out of our total. That makes sense, right? So if we think about the progeny that would be produced by this cross, and again, this parent is only going to be contributing gametes that look like that, right? That's actually our male, not our female. That has to be our male. This is backwards. Females are larger than males. This is what we would expect to get. We could take our 25%, our female contributed gametes here, put them down the sides of a Punnett square, and figure out what we would expect to see in our progeny. So these would be the 4 gamete types coming from mom fly. Dad fly would have only 1 gamete type that he's producing. So this is all 1 gamete. Recognize we have to have 1 of each gene, 1 allele of each gene, and these are 2 separate genes in each gamete. And so in the progeny of this cross, what would we expect? These would be heterozygous at both genes, right? These would be heterozygous at the B gene, homozygous recessive at the VG gene. Whoops, forgot my little underscore. These would be homozygous recessive at the B gene, heterozygous at the VG gene, and these would be homozygous recessive at both. Okay, so those would be the genotypes that we would expect. And what would be the phenotypes associated with that? Well, based on the dominance relationship, these would have normal tan bodies and normal wings. I'm just going to abbreviate normal as NL. These would have normal tan bodies, but vestigial wings. These would have black bodies and normal wings. And these would have black bodies and vestigial wings, right? And we would expect to see these in the following proportions. 25% of our offspring showing each phenotype. So if we generated 1,000 flies as offspring, we'd expect to see 250 of them showing each of these phenotypes, right? 50% showing the phenotypes that were exhibited by our parents: tan body, normal wings, black body, vestigial wings. 50% showing the parental phenotypes, and 50% showing the recombinant phenotype, where we now have tan body combined with vestigial wings, black body combined with normal wings. That's a new combination of traits. 50-50, right? 250 progeny showing each. What Thomas Hunt Morgan observed when he did this experiment was not equal numbers of each progeny type. Instead, what he observed was an overrepresentation of the parental phenotypes compared to the recombinant phenotypes. All right? So what does that mean? That means that we don't have this kind of independent assortment. We don't have independent assortment between the B and the VG genes, which must mean that they are linked on the same chromosome. And the reason that it's less likely for us to have recombinant phenotypes is because those two genes are physically located on the same piece of DNA, and in order to separate those alleles from one another, we have to have a recombination event between them that separates them, right? So the B and the VG alleles are on the same bit of DNA, and the only way that we see a new combination, say the recessive allele with the dominant allele for the other trait, is if there's a crossover between them. And then we would see that new combination of traits that we hadn't seen before: dominant and recessive, recessive and dominant, leading to our recombinant gamete types, and therefore our recombinant progeny after fertilization. All right? So again, how far apart these two genes are from one another is going to determine how many recombination events we expect to happen during meiosis, or how likely it is that we're going to have a recombination event during meiosis. And so therefore, the further apart they are, the more recombination events we can expect to occur, and the more recombinant gametes we can expect to be present. And so we can actually generate a map based on these frequencies, these numbers of recombinants compared to parentals, and comparing the proportions of the two. So in order to do this kind of mapping using recombination analysis, what do you need? Well, you need your heterozygous individual that is heterozygous for two genes on the same chromosome, or that you are hypothesizing are on the same chromosome. And you need to cross that individual, do a test cross, a test mapping cross to a homozygous recessive, right? And then obviously we need to have enough offspring for accurate counts of recombination versus non-recombination progeny, right? The most common classes are going to be represented by the parental phenotypes where no recombination event occurred. The less common are going to be the phenotypes that were formed by a recombination event, right? Now remember, this is recombination happening during prophase of meiosis 1. So what we want to do to generate our map is add up our recombinant progeny and determine what percentage of the total they are. So what percentage of the total are the recombinant progeny types? And that represents a recombination frequency. Right? So using our numbers that we had in our previous cross, we had these black vestigial and gray/tan normal. These were our parental types that correspond to the phenotypes of our original parents. These new combinations of phenotypes are our recombinants. We add those up, get 391, divide by the total, multiply by 100, and that gives us a percentage. This percentage of recombination frequency equals our genetic map distance, or the distance between these two genes in genetic units. And those are referred to as map units, or in honor of Thomas Hunt Morgan, as centimorgans, or cm, to represent those genetic distances. Right? So the closer together two genes are, the fewer recombinants you're going to end up with, and the smaller this number is going to be. So you're going to end up with a smaller genetic distance between those two genes. On the other hand, the further apart those two genes were, the more recombinants you're going to have, the larger this number is, and the greater the genetic distance you're going to determine being between them. All right? Now, can we use recombination analysis like this to map all genes along a chromosome? Well, to a point, but only to a point. So this is going to break down for genes that are far enough apart that we're essentially guaranteed to have a crossover between them. So if we go back to our previous example, if we've got these two genes at opposite ends of the chromosome, and we know that recombination is an obligatory event during meiosis, then we're guaranteed to have a crossover somewhere along this chromosome between A and D, right? And so in that sense, those two genes are going to behave as if they're located on different chromosomes because they're guaranteed to have a recombination event between them. So if we remember back, for independent assortment, genes that are on different chromosomes, we did that, we expect 50% recombinance, right? Under the hypothesis that these are on different chromosomes, we would expect 50% recombinance. So if you have two genes that are more than 50 map units apart, then that's essentially behaving like they're on different chromosomes. So you can only map genetic distances that are less than 50 map units apart, right? Well, a lot of genes are separated by more than 50 map units. So how would you be able to map them relative to one another? Well, you might have to map them sequentially, right? Let's say that these two genes, A and D, are separated by more than 50 map units. So they're assorting as if they're independent, but they're each separated from gene C by less than 50 map units. You could map A versus C and C versus D, and in that way build a genetic map between A and D. All right? So in order to map genes that are separated by greater than 50 map units, or centimorgans, you would have to map them relative to another gene along the chromosome. Okay, so that's recombination analysis. There is a final topic that is optional for you to review. There will be an extra credit question that will be up for grabs on the discussion board, and that's regarding genomic imprinting. So take a look at that video. It's a very interesting genetic phenomenon, but it is extra credit to go over that. And so you can choose to do it or not do it, but I would recommend at least viewing the video because it is a very interesting pattern of inheritance that we observe for a very small subset of genes in the genome in humans, just about 100, and again that's genomic imprinting.